right. Let me share my screen. Oops. All right, are you seeing my screen?
Or you're on the second slide.
I'm on the second slide, all right. Well, hopefully I'm on something. Okay, so I'd like to go ahead and welcome everybody today for our fifth and final session of the Introduction for Plant and Plant Microbiomes Learning Sessions. And again, this work has been put together by myself, Deborah Wells, and Charles Quincy, and the work is funded by the NASA Space Biology Program. So why are we here today? So NASA's facing a huge challenge, and that challenge is that the astronauts have to be far more sustainable in order to stay at places like the Moon and Mars at these long-duration space missions. And again, this is such a big challenge that NASA is looking at this in different ways. One approach is we'll be launching a centennial food challenge on this in January, where teams will come together and compete in order to design sustainable food systems for astronauts for the Moon and Mars. The other way is to try and understand the science that underlies the biology that goes on in deep space. And so what do we need to do in order to understand that science? Well, the first thing we need to do is understand what knowledge gaps that we have. And we need to have a good community that will be able to both outline what those gaps are and then also to fill those gaps, and then to outline what the opportunities are going to look like. And in a broad way, that's what this series of talks has been about. The community that we've put together is a group of space scientists, plant microbiome scientists, space platform experts, science hardware designers, space environment experts, and then people working on cutting-edge science tools. And we've gone through all of the talks now except for the space platform part, which is why I left that out of today's introduction. And today, Debbie Wells will be speaking to us about that. So again, we've done a series of four talks so far. Today is the fifth talk on space platforms. And really, these are about trying to get our community to understand what the challenges facing other members of the community are. This will culminate in a series of two workshops. The first workshop will actually be held down here on November 17th. It will be the plant research workshop as part of the ASGSR decadal survey preparation. So you can go to asgsr.org to register for that workshop. And the second set of workshops will likely be happening in January, and we'll send out information on how to register for those. The workshops that will be organized around developing white papers. The first set is going to be around developing science white papers, and the second part will be about the transformational hardware that's going to be necessary to help do the science that we want to do. So with that, I'd like to turn it over to Deb Wells, who will be doing the space platform and space environments for us today.
Thank you, Bruce. And welcome to our participants today. I'm going to share my screen. So give me 1 second.
Looks like you're sharing.
All right. So for anyone who hasn't met me before, I'll just I'll tell you a little bit about myself. I'm going to also turn off my video. Give me 1 second because it looks like it's using a bunch of bandwidth. Okay. So I'm a biomedical engineer. I've been working here at Kennedy Space Center for 35 years or so. I spent a little bit of time away from the Space Center in the entrepreneurial space and venture capital, but primarily been working in space life sciences since the beginning, 1988 or so. I'm currently the deputy program manager for a contract at Kennedy Space Center called the Laboratory Support Services and Operations Contract. And part of what we do is to support NASA's plant space biology program here at Kennedy Space Center. And so I've been working with Bruce and Charlie on these seminars and the workshops that are coming up so that we can be prepared for what's coming as we move to the moon before we go to Mars. So that's really the title of today's talk, is to talk about what are the platforms that are coming up in the next 10 years that are available to the community. And so that you know what to expect and how to access those platforms and how to plan for them. So I think many of us heard this direction from Jim Bridenstine, our NASA administrator, that our goal is to put the first woman and the next man on the moon by 2024. And so a lot of, in fact, almost everything that NASA is doing right now in terms of human spaceflight is really focused on meeting this goal. So all the things that we're going to talk today about have to do with meeting this goal and how we can leverage that to move forward the program in, hopefully crop production in space. So I think we're all familiar with, but I'm going to go over these real quick again, the 5 major categories of hazards of human spaceflight. One is radiation. And that obviously has a meaning to vehicle systems and humans, but it also has a meaning to the plants and the other living organisms that go along with that system as we go through space. Isolation and confinement, psychologically for the crew members, that means a lot of things, but it also has impacts, again, to our living systems that we need to think about in terms of, circadian rhythm changes and even just, the difference in environment. Distance from Earth means we can't communicate as well, potentially, or as frequently. It means that sample return is a little more complicated. And it means we have to do more things where we are, more analysis, and being able to send back data. That lack of gravity, or maybe we, want to say variable gravity, because as we do our exploration missions, in microgravity, in orbit, landing on the surface of the moon, and then finally on Mars, it's really about handling that variable gravity. And then finally, those closed environments of the spacecraft are not like our laboratories and the Earth here where we typically are working. And so there's a lot of changes there that create stressors in our living organisms that we're interested in plants and the microbiome. So what does that mean? So radiation, we have to worry about those, double helix strand breaks and other damage to those core fundamental mapping that produce our organisms and how they operate. And, so that affects gene expression, but gene expression is affected not only by the radiation, but also by all those other stressors that we talked about in the spacecraft, about the changes in temperature and pressure, and even the changes in gravity. And those, we know our organisms are adaptable, but with the variable environments we're going to see over the course of these deep space exploration, you're going to have change you're going to have stress and adaptation and stress and adaptation. So it's going to be a cycle. And then there's that, getting samples back. How do we collect information and bring it back when we're at a remote location? And water handling, the microgravity effects of being able to control water and put it where you want. I think we've all seen these photos of a water droplet on the leaf of the plant, making a really nice ball. It looks very controllable, but you also notice in this picture where the astronaut's hand is in the water that the water wants to cover that entire surface of his hand. And so this creates real issues for us with growing plants and being able to get the water to be where we want it to be. And then obviously the microbiome, you guys have heard the talks over the last sessions about the changes in the microbiome and what we're seeing just on the early space flight experiments that we've done so far. So what are we really trying to accomplish? We're trying to find those answers to those questions, to those hazards. And we have various platforms that allow us to study those different things. We have the ISS, we have analogs, and we're going to talk today about what do these other platforms look like. Gateway, what is the lunar surface capability, and when we're traveling there and back, what does that all look like? And this is, part of the picture is, when we're working here in low Earth orbit on the left, we're close to Earth, but there's a certain set of parameters. It means we can do more as we go to the moon's surface, as we go to the lunar orbit. We're still close in terms of Mars, but it gives us a little bit further away. It gives us the opportunity to explore communications and what we can do in terms of crew time in that type of environment before we move to Mars. So Bruce and Charlie and the team at KSC, we kind of put this chart together to try to show, how do we study those different hazards on the left-hand side and all the different environments that you're going to see and the types of research that can be done on each one. So you have here on Earth on the left-hand side with analogs, moving to hyper-G and parabolic and other analog opportunities to study gravity changes. We have the ability to study microgravity changes on the ISS and on other platforms in LEO. And then as you move into the programs and platforms we're going to talk about today, you can see that we can expand those hazards. We can talk in terms of studies on radiation, magnetic field, and then the duration of those experiments as well. So by using all of these platforms in the right way, then we can study all of those hazards and be ready to provide solutions for when we're ready to go to Mars. So in terms of analogs, I think many of you would be familiar with these. On the left, on the top, is a piece of equipment called a synthicon that a lot of researchers use here on Earth to simulate microgravity conditions in cell culture. We also have just below that the hyper-G centrifuge available. We can study radiation in some small way using the Brookhaven National Lab and the beam that they have there to expose samples to a certain level of radiation and then observe the changes. We're doing quite a few experiments with this group and Kennedy Space Center using seeds, exposing seeds, and then growing the plants to see what the changes are. We also have environments like the Eden ISS, which is actually not the space station, but it is actually into Antarctica that gives you a remote location for crop production and the effects of that closed kind of hostile environment and what it takes to operate in that type of an environment. And then also you have opportunities like the Blue Origin suborbital program, where we can test not only technologies, but also research as well. So these are all opportunities here on Earth for analog research. Then as we move out into low Earth orbit, we have the International Space Station. You guys have heard presentations on the advanced plant habitat, which is on the left, the veggie, which is on the bottom right. The new piece of hardware called Spectrum, which gives us a robust imaging capability now on the International Space Station. which Dr. Paul talked a little bit about last week. But I also wanted to mention that, we do have these CubeSat opportunities as well in low Earth orbit. Ames Research Center has flown several of these where we have an opportunity to study biology in a CubeSat type system. So small, highly repeatable type experiments in low Earth orbit, just not on the International Space Station. So coming up on the International Space Station, you're also going to have an opportunity to test hardware and do research in a system that's called the Ohalo 3. This is a test platform primarily designed to test watering and lighting and other systems that would help us test and demonstrate their ability to support a crop production, so a larger scale crop production as we move into the future for, say, lunar surface operations or even Mars surface operations. So this hardware becomes available in low Earth orbit again within the next couple of years. But what we're really here to talk about today is, the step 3, which is moving beyond low Earth orbit. And what can we do there? So in NASA's vernacular, the moon before Mars is our priority. And this is where we're going to spend the rest of the time today talking about those platforms and what's available. So down in your multimedia viewer that's part of the WebEx system, hopefully you guys can pull that up. There's a video queued up for you to watch at this point that kind of gives you a nice overview of what NASA is planning. So I'm going to give you a moment to find that window, the multimedia window, and pull that up and watch that video.
Deb, do you want to just share that to the multimedia window again?
I sure can. It's there now.
There. And so folks, you need to click continue and then the video should pop right up. Thank you. All right, I think the video's over for most people.
Yep. I'm just going to pull up the presentation back where we were.
So you need to stop the video.
Okay, give me 1 minute.
Sorry, that was my video.
Yeah, everybody should be able to stop it themselves.
All right, so I think you're good to carry on.
Okay, thanks, Bruce. Try to get back to the slide where I was.
And while Deb's doing that, I just want to say that we'll send out a link to the video in case you did have a problem with that.
Thanks, Bruce. All right, so that video was a nice overview. When I watched it, really helped me to understand sort of the logistics. You've got the pre-positioned payloads. If you remember, it talked about that on the moon. So some of the resources that we need will be pre-positioned. And one of the ways that NASA is doing that is through their commercial lunar payload systems, the CLIPS solicitation and the payloads that are chosen as part of that. And so this is really the foundation for our human missions. And the first of these start actually next year. And it includes landers. So these are, hardware that land and stay in one spot, but it also includes a rover called the Volatiles Investigating Polar Exploration Rover, Viper. So the Viper rover is also part of that initial program. And I wanted to point out here that NASA is asking for community input into what experiments should be done in this area. And on this program. So they had a stage 1 request for information. It was opened and closed, and you should expect to see additional solicitations and RFIs, requests for information around what we should do next. And then those would come out as part of a research solicitation as well. So things to contribute to NASA's understanding about what the community thinks is important, but also how to apply for one of those grants and contracts. So I want to just mention briefly, one of those landers is being provided by a company called Astrobotic. And this is their peregrine lander. And to give you a sense of the accommodations for an experiment on this lander, there's a photo of it on the left. You can see it's got a solar panel. It has some communication capabilities. It would obviously land on the moon. And the idea is that it would have about 192 hours worth of operations. Any experiments are basically going to be in a box that's mounted on these little mounting decks. And the mounting decks have
Debbie, can you hang on a second? People can't hear anything except the video, so apparently it's still playing somewhere.
Okay.
If that's, wondering if that's me.
Let me see if there's a way for me to turn that off. Give me 1 second.
Maybe you can unshare the, let's see. I'm not finding my controls from my multimedia.
I had that same problem, and if you disconnect from the player down in the right-hand corner by hitting the X, it turns off.
Thanks, Charlie. Yeah, there's no way for me to turn it off from here. I think each person has to do it themselves.
I'm just going to put that in the answer.
Okay. I'm going to go back to the presentation then.
Charlie, can you tell people in the Q&A that?
Yeah, it was posted in the chat as well.
Okay, good. All right, with that, then let's go ahead and continue.
All right. Technology is a wonderful thing. Okay, so the payload accommodations on this lander, just to give you a feel, it's about a half of meter squared, and it says per side. So if you're looking at this mounting platform, what that means is the top side is about a half a meter squared, and the underneath the bottom side. So payloads can be on the top or on the bottom. So this is kind of like the little porch on the side of this lander, but that is basically the size of what is available for payloads. The total mass of payload for the whole lander is about 100 kilograms. So that gives you some idea of, the size and possibilities for a payload on one of these CLIPS landers. So on the first Orion flight, so this is called Artemis 1. The first Orion flight is gonna leave Earth. It's gonna go out around the moon and come back. And so we actually have 4 biological crew compartment payloads. So they're going to be inside the habitable volume of that first Orion flight. They don't have power. They do have habitable conditions. Everything has to be self-contained, and the experiment hardware will come back. And so most of these experimenters have chosen pretty simple systems, either petri dishes or tubes, conical tubes. One particular experiment is using a piece of hardware that was developed for the ISS, more of a culture box. So there's 4 investigations. Dr. Brandisi is studying plant seeds, Arabidopsis. Tim Hammond has an algae that he is looking at. Dr. Wong is doing a fungi experiment, and then Dr. Zaya from University of Colorado Boulder is doing a yeast. And I'm just going to show you a little bit about his hardware so you get a better feel for, again, what's possible on the on this Artemis 1 flight, and is likely going to be similar when we go to the Artemis 2 and potentially Artemis 3. So this is a small box. The size is about 4 by 10 by 7 inches that he has his yeast experiment in. And the hardware is called the Peristaltic Laboratory for Automated Science with Multi-Generation. So you can look that up. It was used for at least one other experiment. And the interesting thing that they're going to do here is they're going to have one of these boxes on the Orion for Artemis 1. They're going to have 2 on the ISS and then 2 on the Earth. So they've got the same hardware in the multiple environments so that they can compare the science between those different platforms and between those different conditions. If you remember at the beginning, we talked about the hazards that we can study in those different platforms. And so you're able to matrix all of that information together to really start to tease apart some of these questions that we're asking. And I put Dr. Zaya's website here so you guys can follow along on this experiment and see how it progresses. They're actually in the middle of what's called the experiment verification test here at Kennedy Space Center right now for all 4 of these experiments. So they're on progress to launch with that first Artemis 1 flight, which I believe is toward the end of next year. So the secondary payload, so what we just talked about was payloads that are in the crew compartment on that Artemis 1 flight and kind of the size and shape, capabilities, resources that are available to them. But there's also an opportunity here in the service module for payloads that can be deployed during this flight as well. And so these are called the secondary payloads, and they're about the size of a 6U, in terms of CubeSat, about 6U size, so 4 by 9 by 14 inches, no more than 30 pounds. And basically you have to be, again, completely autonomous. And Ames Research Center is actually launching a free flyer that will go out into a heliocentric orbit, a biosentinel payload that'll begin to collect data on that deep space radiation. And you can see one of the little wells plates that is being used. And this is again studying yeast as it goes out into that orbit and comes back. So it's using an LED detection, 3-color detection, because these samples won't come back. So they're relying on that communication of information to come back for the data for this payload. So again, Artemis 1, you've got opportunities in the crew compartment as well as in this secondary payload area, more of a CubeSat type possibility. For Artemis 2, we know we're going to have secondary payload opportunities again out here in that stage adapter area. These are going to be larger, potentially 12U and even 27U areas to be deployed on the Artemis 2. We're not sure yet. We haven't seen whether or not there's going to be space available inside the crew compartment like there is for Artemis 1. This flight does have 4 crew members planned to go with it. So you've got, food and, all the other associated items for the crew, which may limit our accommodations inside the crew compartment. But if we do have those available, they're going to be similar to what you saw just a minute ago for Artemis 1. So right now, there's not a solicitation released for the Artemis 2 crew compartment payloads, but as I said, expected to be similar to Artemis 1. So the video also mentioned Gateway. So Gateway, I want to tell you a little bit about that. So this is a system that will be in an orbit around the moon that allows us to dock with it. Orion docks with it. The HLS lunar landers dock with it. The crew can live in this for a certain time, sort of as a transition point. And the habitation part of this is called the HALO, Habitation and Logistics Outpost, and Northrop Grumman has been awarded the contract for that. It's based on the Cygnus platform. So those of you guys that are familiar with the current ISS commercial resupply are familiar with Cygnus. This is one of the platforms that we currently bring experiments to and from the space station aboard. This platform has been used to bring live rodents to the ISS. And is intended to act as laboratory space. And in fact, it is separated from the station and operated for over a year as a sort of low Earth orbit platform as well. So it will be enhanced to provide better command control, environmental, and it'll be able to sustain 4 astronauts for up to 30 days is the plan. So along with the Gateway HALO, the habitation module, there is what's called a Gateway Logistics Services module. So this has been awarded to SpaceX. So this logistics module flies from Earth to the Gateway and back. So it gives us a way to bring supplies, whether it be for the crew or whether it would be scientific supplies and materials back and forth between the Gateway and Earth. The only other way to get things back and forth between the Gateway and Earth is the Orion capsule, which we know is going to be primarily for crew and crew support materials and critical science samples, however those are defined. So this right now is based on the CRS Dragon logistics module. So the logistics module that we currently use to go back and forth from the space station. However, it will be much larger, and the plan right now is that it would be launched on a Falcon Heavy rocket. So obviously a lot more capability, a lot more storage in this system. However, this has been taken out of what we call the critical path for our first mission to the lunar surface. So until we get past Artemis 3, which is the first time we land on the moon, we can't expect that we're going to have access to this to get anything back and forth from the gateway or the lunar surface. We're going to be primarily reliant on the Orion vehicle to bring things back and forth. So Artemis 3, this is the big flight. This is the 2024 goal, to land the first woman and man on the lunar surface. And so you've probably heard in the news, there's 3 different vehicles currently in competition to be that system, to be that human landing system. Dynetics, SpaceX, and Blue Origin each have their own concepts. And just to kind of bring you up to speed, Dynetics has delivered a test article to NASA. It's in Huntsville. And, it's really there for the crew to be able to work with engineers, to be able to look at the astronauts, to get a feel for, the habitability, where systems were, is everything in the right place? Get a feel for that before they move forward in the design. Blue Origin has delivered an engineering mock-up. Same kind of idea here. The astronauts and engineers will be able to do some testing and validate the systems. This one's at Johnson Space Center. And SpaceX is working on theirs at Brownsville, Texas. They're keeping fairly quiet about the status and capabilities, but we would expect to hear more. All 3 of these are in competition, obviously, for the final selection. So what does that look like? What is that Artemis 3 flight going to look like? First of all, the current concept of operations leave room that the gateway may not be used for that first flight. So we talked about the logistics module not being part of the first flight, but it's even possible that the gateway itself may not be part of that operation. So we may not see gateway and the gateway logistics module until after. So they're preserving the ability for the HLS and the Orion to dock directly or to use gateway at this time. So the amount of science capability changes greatly whether or not gateway is available in this time frame. So again, we've got to keep thinking, small resource limited possibilities for science. We also have to keep in mind that the atmosphere aboard the HLS, when it's leaving Earth and going to the orbit around the moon, it may not be a habitable environment until they prep it for the crew to interface to it. So the conditions in these vehicles, both the HLS and even the gateway, when there are no crew there, may not be kept at a completely habitable situation. So you may have lower pressures, you may have wider temperature, humidity, those types of things. We may see those changes. However, obviously, once the crew is aboard, you would have more standard habitable conditions. And the idea is that 2 crew would always stay on orbit, while the other 2 would go to the lunar surface on these first missions. Artemis 3, the very first one, is looking at about 220 pounds of science tools and equipment to the surface using the HLS landers. The idea would be to have about 4 EVAs over the period of the 7 days while they're there to do work. With the primary goal in this first mission is geology, especially in those permanently shadowed regions. We're trying to get a better feel for what's available on the moon's surface. There's a lot of talk in the news just in the recent days, even about water. Day 3 would be a day of rest, and I put that here because this is an opportunity for them to do science inside the ascent vehicle, so within the habitable system, and even including some public engagement. So again, another good opportunity, not only outside on the surface activity. So, maybe you've landed a particular science payload on one of those CLIPS landers on the outside platform. It finishes after the 192 hours, and maybe these crew members can go over and retrieve some of that science and bring it home, or have a visible data collection that they do. So things to think about in terms of what's possible at these times. Artemis 4 and beyond, currently the plan involves a foundation surface habitat of allowing about a week for 4 crew members to stay, a vehicle to move around the moon surface, and even a vehicle that is that you can move around and has a livable habitat on it as well. And at this point, Gateway is available. It becomes that true transition point between Earth and the lunar surface. And again, expanding our ability to do science that looks a little more like what we see on the International Space Station. So this timeline gives you an opportunity to see, mission duration, how many crew are involved, days on the lunar surface, as we move through the Artemis program, and what's available in the next 10 years or so as far as these payload opportunities. So trying to wrap things up, so I leave you all time for questions. We've talked about a lot of different possibilities for science over the last 4 seminars like these. Today, we talked about platforms that are available for doing that science. We talked about analogs on the Earth, where we can spend more time, ask bigger questions, have a longer term research program. We've talked about low Earth orbit, not only the International Space Station that allows us to prove out technologies and demonstrate solutions that we've come up with, and we still have multiple opportunities there. But that, keeping in mind that we have that free flyer opportunity in low Earth orbit as well. If anybody is interested in science that you can do on the CubeSats in these heliocentric orbits, or even coming off one of the Artemis flights to go farther into space, if you have the same hardware for a free flyer in low Earth orbit that then you would then use in those deeper space missions, you get some repeatability and some extension of research from one platform to another environment. So between the Earth and the Moon, we've talked about small satellites like the BioSentinel, where you can ask very focused questions, but you have challenges because you have no sample return. You have the CLIPS landers. Again, some limitations. You have, limited durations, no environmental control, but it gives you an opportunity to test lunar G, radiation, and to verify things for surface operations as well. Artemis 1 and 2, this is really, between the Earth and the Moon, small scale. You saw the kind of size of Dr. Zaya's piece of hardware. A limited amount of volume is going to be available there, and probably very limited power, communications, and crew time for these flights. But remember, you also have that secondary payload opportunity if you have a CubeSat type activity. We're keeping in mind that those opportunities are going to get larger between Artemis 1 and Artemis 2, up to 27U possibilities. And then finally, on the lunar surface, Artemis 3, that very first flight, it's going to be pretty constrained. There's going to be a lot of goals around geology, and it's very dependent on whether we have Gateway in the system or not, the volume and mass that we can bring with us and then bring home. We really have a lot more opportunities when we get Gateway up and running in the Halo, that again will look very much like an ISS, probably very much like ISS did, in fact, in the early in the early months and years, we had limited science capabilities, but after we, finalized our concept of operations and were able to finish building and getting systems up and running, then you had more of an operational opportunity there on ISS, and Gateway is likely to look similar. And then finally, you've got the base camp. Keeping in mind that, again, NASA has some very specific goals here for ISRU and being able to demonstrate some of those things. But there will always be opportunities, especially for these smaller payloads, and especially anything that can use communication to get data back versus sample return in these platforms. So a lot of possibilities exist to do studies on these various platforms to try to understand those hazards that we talked about at the very beginning. So the real question that all of these seminars is trying to get you to think about is, how do we use these platforms to not only understand, so the science, the fundamental science, but really to demonstrate that we can have safe and nutritious crop production as a part of our exploration strategy. So I hope that this helped today to bring you at least a little bit more aware of the various platforms, the various opportunities, the constraints that have to do with these platforms as we move forward. So I'm ready. Are you?
Thanks, Debbie. That was a great talk. Charlie, I don't know if you've got any questions that you can see that you are ready to ask Deb.
One of the questions was on the pressures on the gateway, and I think that's still a little bit of a TBD at this point. But the requirements for sustaining the gateway is very important, and we need to make our inputs to that group to get as close to what we want to have as possible. Otherwise, they'll design it to be completely safe for the gateway, but maybe not all that good for biology.
Yeah, there's it when I was doing the work to prep this, Charlie, in talking with both the folks involved in the different programs, it's really going to depend on the final designs of the HLS systems, the final designs of the gateway, and their usage of the available supplies to maintain a certain environment. They're obviously going to want to maintain an environment that preserves the ability of the equipment and everything to function and be ready to come, up to a habitable condition when the crew is there, but conserve resources when the crew is not there. So I think that that particular number, is it going to be 10.2 psi? Is it going to be something less than that? Is something that we can't put our finger on at the moment.
Right. And there's been a number of questions today on getting copies of the slides and getting access to the presentations we've made over the last 5 weeks. The plan right now is to upload the slides to an available website, and as soon as we get that done and to everybody that has actually linked into the talks, and we'll send it to everyone that's linked in, even if you weren't on at all the talks.
So Charlie, I'm not sure if you're dropping out or not, but Charlie said that we're working on getting those uploaded to an available website, and I'm hoping that we'll have that done within the next week, actually. So I think I've got almost all the approvals I need to do that at this point. There's one person in the Q&A is pointing out, Debbie, that there is a sample return from small sats is now possible. I think they're with orbital transports. That's probably from CubeSats in Earth orbit, probably not from translunar or solar orbits, I would guess. But we could check.
Yep, that's a good point.
So there's another question here about what plant growth systems will be available in this architecture.
And I guess we should thank Dr. Sugar for that question.
Yes, we should.
So it's a good question. I mean, we're doing things on the ISS today. So the systems like Veggie, advanced plant habitat, and even the new ISS test rig that I mentioned are have all demonstrated our ability to do plant science at different levels. And we've done even smaller things like using the magenta boxes to grow plants in. The Arabidopsis experiment that's on Artemis 1 is just going to be using a conical tube to grow plants in. So I think our challenge is something like, veggie, APH, advanced plant habitat, and even a full crop production system isn't going to fit in any of these platforms until we start talking about a surface habitat at probably anything larger than what we've seen on the ISS. A veggie could translate pretty easily to gateway and even an advanced plant habitat. The gateway is also designed to be expanded in some of the pictures that you see out there on it, and some of the discussions about it is, it could be expanded with something like a Bigelow module, the expandable modules that would give you quite a bit more volume to do larger things like the ISS test rig, and transfer that out to gateway and then really do a better crop demonstration. So there's a lot of opportunities with what we have, but part of what we hope to get out of one of these workshops as we go forward is what should that hardware look like on these various platforms, and wouldn't it be nice if we had a piece of hardware that could be on all of those platforms so that you could have the same science in all of those platforms to really be able to answer all of those questions at the same time.
Very good, Debbie. If I could expand on that just a little bit also, there was a good deal of talk, 2 or 3 years ago on a gateway garden. That was pushed back by the technology people because the need for food production capability at Gateway could not be all that well supported, and maybe even for the lunar surface. But as we go forward and demonstrate the capabilities to go to Mars, food production becomes more and more important because of the nutrient decay that goes on over time and the length of time of those missions have. So the short answer is if you can come up with a good demonstration product for Gateway and the lunar surface, I think there's a high probability of keeping them in place. To get to a production capability at this point, maybe a little further down the line when a more permanent settlement on the moon happens.
And I guess from my perspective, is think small. Obviously, as you think about going to the moon, mass and power are huge restrictions for these missions. So if you really want to get onto some of these missions, the smaller you can make that design and the less resources you use, the more able NASA is going to be able to fly that. And if the quality of the science coming out of those packages is high, that's what's going to push them up the list to be selected for a space flight. And the talks I've been sitting in, or have heard a little bit about anyway, discussing Gateway and lunar landing, we're just not going to have the space for a long time onboard gateway or the lunar landing vehicles to do a lot of stuff. Even if we do veggies, it may be a bit smaller than it has been on the ISS. And certainly we're not looking at sending anything like the advanced plant habitat to either gateway or the lunar surface at this point in time. So, you know, think small and think good science. So, yeah, I see a question here about fern spores. I think that's fine. That would be a fine system, as long as we're getting the kind of, you know, it's the science you're getting out of that. If you're getting good science out of that, then that will help a lot. There's one question that comes up. I don't know if you have an answer to this, Debbie, and it regards, I think, Planetary Protection Act and bacteria going to the surface of these planets, probiotic or whatever. Do you have any response to that?
Yeah, I saw that question, and there was an updated policy on this that came out, and I would have to go look at it. So I was going to ask about we'll answer that question when we post the answers to the Q&A, because there's definitely some updated guidance in that area.
Yeah, that's probably the best thing to do with that one.
I will mention that NASA does have some pretty outstanding information out there on the Artemis program. There was a new document called the Artemis Plan that was put out, and a lot of the companies that have already been selected either for HLS, for the Gateway, they have what they call Payload user guides. They have mission planning guides that are out there, pretty easy to find. The information is more intended for the larger payloads, but like for the CLIPS landers, you know, Astrobiotic has a good set of information on their website. Not only you know what the capabilities are, but they're also, you know, looking for commercial partners to fly on these landers. So you know, the only way to fly and ask these questions and answer these questions is not just through NASA solicitations. You can also have commercial partners in these areas and fly together, you know, as an independent payload on these platforms as well. So, you know, just, you know, think, you know, differently. It's not all just what NASA is soliciting either.
Great. Thanks, Debbie. I'd really like to thank you for a great talk today. It was certainly pulled a lot of things together. I wasn't nearly aware of the cube satellite type storage space that will be in the adapter rings and so on for Artemis. It was really good to see that information. At this point, I'd like to go ahead and end the meeting because we're at 1:02, and I'm sure that Debbie has other things she has to get to. So thank everybody. I'd like to thank everybody for attending these learning sessions, and we will get these posted, and I'll send out an email as to where to find those, hopefully here in the next several days.
Thanks, everyone. Thanks, Bruce.
You're welcome. So I'm going to go ahead and end the recording.
Thanks to all.